A motor neuron meets a muscle fiber at the neuromuscular junction. The action potential travels to the end of the presynaptic motor neuron. This then causes the release of the acetylcholine neurotransmitter into the synaptic cleft, where the neurotransmitter binds to acetylcholine receptors on the muscle fiber. This then opens sodium channels to depolarize the muscle fiber, resulting in an action potential being generated. This action potential is propagated along the sarcolemma.
Action potential travels through the sarcomere and down the T-tubules until it hits the sarcoplasmic reticulum. The sarcoplasmic reticulum then releases calcium ions.
Calcium ions travel into the sarcomeres, where it binds to troponin, which essentially moves trypomyosin out of the way to reveal actin's binding sites. At this point, ADP and phosphate are attached to the myosin heads as a result of the previous ATP breakdown. The energy is stored in the myosin heads. It is in a cocked position, and now that the binding sites on actin are opened, the myosin heads will bind to actin, forming cross bridges. At this point, the phosphate leaves the myosin head. Using the energy stored in the myosin head, the myosin will pull actin across itself, almost as if it is walking across the actin, and in this process, ADP is released. To release the myosin head, ATP is used to break the bond between the head and actin, relaxing the myosin. ATP is then broken down by an enzyme ATPase, and energy is released back into the myosin head, and it is in the cocked position once again, and ADP and phosphate are once again on the myosin head. If calcium is still present and binds to troponin, the cycle will repeat until there is no more calcium.
The process just described is happening in multiple layers, all in many different sarcomeres. Here is one layer of a sarcomere. As the myosin heads continue to pull the actin closer to the M line, the sarcomere begins to shorten.
When breathing and circulation stop, muscle cells lack oxygen and therefore cannot use aerobic respiration to efficiently produce ATP. Respiration continues anaerobically at first, but the muscle cells eventually become so short on ATP that the myosin and actin filaments cannot release from the contracted state, and the calcium ions cannot be pumped back out of the muscle cell. Unable to release contraction, all the muscles of the body remain tense, causing rigor mortis.